So, good afternoon, everybody. To most people's disappointment here, I'm not actually going to be talking about insects today. I'm going to be talking about mites. But I'm sure Dean Reddy would love to hear about how I'm a psychologist of mites. Today, I'm going to be presenting my work on how the application of microbial pesticides can change mite behavior. So, first I'm going to give you a brief introduction about biopesticides and volatiles. Then I'm going to go over my objectives, methods and materials, results, and of course my conclusion. Because who wouldn't want to hear that? So who knows what biopesticides are? Has anybody heard of them? Right there? Yeah. Well, that's unfortunately not many of you. So biopesticides are anything that may be made out of a plant product, animal product, mineral, or microbe. So how many people have heard of BT? There we go, that's a lot more hands. Okay, that is a biopesticide. So because of the organic movement from the environmental movement, just as Dean Reddy was saying earlier, everybody wants a healthier environment. So we're getting an increase in the organic foods, and therefore biopesticides are often used in the organic food market. It's increasing right now about 10% annually, and in 2022 it is expected to go up to $8.8 billion. So I want to mention too that plants communicate through volatile organic compounds. When a plant is attacked by an herbivore or a pathogen, it can have secondary metabolites that increase either toxic compounds, or also the plant can release volatiles that can be a deterrent to pests. It can attract even the predator of those pests. Or it also is like a warning signal to other plants to prime their immune systems to get ready for attack. So I worked with ornamental pepper because it's somewhere in between an ornamental crop, of course, but also very similar to a vegetable. So I figured that this would kind of go across the board to be a good figure model. And I'm sure if anybody here is working in a greenhouse, they know how annoying two-spotted spider mites are. They are a huge problem in most greenhouses, very annoying, and they feed over 150 different species of plants. So in summary, what we know is that organics are becoming very popular. And because of that, there's also an increased use in insecticidal and fungicidal microbes. But what we don't know is what are the effects on two-spotted spider mites. So these are the microbes that I use. My foliar products used specifically for two-spotted spider mites were Grandevo and Ancora. And then my soil products, which were intended for Phytophthora capsicum, were RootShield Plus and Stargus, which were soil drenches. And the reason why I used those fungicides is because you don't just have one pest in the greenhouse, you're going to have multiple pests like in any natural ecosystem, or even man-made ecosystem. So these are the microbial pesticides at the rates that they were suggested to be applied at. So my pepper plants were propagated from seed in normal greenhouse conditions, and we used a random control block design with 2 trials and 10 reps. So my objective 1. Question is, what is the behavioral response of spider mites on leaf discs treated with microbial products compared to a control? So I set up a choice test first, using 2 control leaf discs. One had been treated with the foliar or soil microbial product, and the other one was just simply a piece of a pepper leaf. And then we set the mite in the middle and tried to see if there was any behavioral difference, if it chose maybe the control over the treatment, if there was any deterrent. That's what we were looking for. So I used EthoVision 14.0, which is basically a fancy video system where we record the mite and then see their behavior. So this is a little video. Let's see if it'll work on a click. Nope. So this is a little video of EthoVision. That red dot right there is the mite. And you can see how it's moving around on the petri dish, going to one leaf and then the other. And the red line shows its velocity and direction. So we also used heat maps to determine how long the mite spent in an area. So in the first figure, we see a preference for a treatment. As far as the second figure, we see a preference for control based on the red coloring. So the next, we were asking, are spider mites attracted or avoid plants treated with microbial products? We use SAS to evaluate our stats and use PropGLM. So we found that the average duration on mite versus a treatment did not change throughout the board, but we did notice that Ancora had significantly less visits to either of the leaf discs than any of the other products. So this just shows there's no difference in location at everything else except for Ancora. But here we can see that they were on the rim significantly more than on either of the leaf discs. This suggests a possibility of avoidance from possibly the Ancora. That's still yet to be decided. So our second objective, another good question, is will the spider mites lay eggs on treated leaves? So we set up floral foams in a square with a petri dish, and then filled the bottom with water so it's a little island where the mites can't escape. We used a cork board to make a leaf disc and placed it with the bottom side up, and then we put 2 female spider mites on each of the leaf discs. We then incubated them in an incubator at 25 degrees Celsius, and then we evaluated them 48 hours later. And I wanted to mention that these did have the tops on, so no mites were escaping. They were forced to lay eggs on that island. So we found that Grandevo had a significantly less number of eggs than any of the other treatments. The averages, these are the total averages by day. I took them on day 5, day 14, and day 34. You can see here that on day 5, there was no significance in Grandevo compared to the others, but come day 14 and day 34, there was. So in conclusion, there is no preference for or against any of the discs treated with these products. Though Ancora shows signs of avoidance, and leaves treated with Grandevo showed significantly less eggs. There's still unanswered questions though. So our future plans are to find out why does Ancora show signs of avoidance. So we're going to analyze the volatiles produced by plants that were treated with these different microbes. And then also another question from our second objective is, are there less eggs on Grandevo because the mites are dying? And so we're going to do direct and residual treatments to find out if that's the reason why they're not laying eggs. And I would quickly like to say thank you to my advisor for being awesome. And then also my other grad students, Axel and Vivek. I couldn't have done it without you guys.
Thank you.
So are there any questions?